Yeah, I will start the class. Good evening.
Good
Yesterday we are
evening,
discussing
sir.
about yesterday we are discussing about DNA sequencing. Like we have discussed how we can sequence the DNA with the different platforms. We have discussed first generation sequencing, next generation sequencing, and third generation sequencing. And we have just looked about the overall summary of how NGS workflow. Like we're going first we'll receive the raw data, then we are going for quality check. Then we are going for trimming. Then after that we are going for alignment. Then we having variant annotation and data processing for further uses. This is the overall protocol of your sequencing. Today we are this is about yesterday we just discussed about how sequencing will happen. Today we are going to discuss some of the how we can sequence RNA and how we can do sequencing of RNA, then how it can be used for gene expression analysis. Today, we're going to discuss about that. And before going to that, we should understand what is gene expression analysis and what are the different techniques are available for gene expression analysis. And today we are going to discuss that. For that, we know that central dogma of molecular biology, where the information present in the DNA is transferred to RNA, then it is transferred to protein. So that the transcriptome is nothing but like what is the transcriptome? Nothing but the total RNA content in the cell at particular time and at particular site at particular cell. Okay, it varies from, for example, you are taking RNA from roots and you are taking RNA from plants. It's totally different. And you are taking the RNA from root at 2 days into 2 days after germination, and you take RNA after 4 days after germination from the same root cell, it will be different. That's why when you're defining transcriptome is transcriptome is the sum total of RNA present in a cell at particular time and particular site. Okay, within a cell also sometimes what happen, then at different site there will be different RNA. That's also nowadays it's coming like single cell transcriptomics. But now today we are going to discuss only the bulk transcriptome, like transcriptome, to entire transcriptome of a overall cells. Okay, then how we can analyze the transcript? We studied about transcriptome. How we can study what are all the transcript are present in a cell at particular time. The study is called as transcriptomics, means study of transcriptome at particular time and at particular cell at a particular time point. This is called as transcriptomics. Then how we can study transcriptomics? There are n number of approaches developed time to time. For example, when you look, normally, like we normally consider it's a part of functional genomics. We have discussed like what is functional genomics, what is structural genomics, what is comparative genomics. Okay. From the structural genomics, the techniques are used genome mapping, genome sequencing, and genome annotation. Yesterday we have discussed in detail. Then what other techniques are available to study the RNA? That are the RNA-seq method is there, Northern blot is there, qRT-PCR is there, and there are several other technologies. Like SAGE was the one technology, serial analysis of gene expression, and there is a another technology called as ddPCR, like a differential display PCR. There are several other technologies, but majorly used till now today is qRT-PCR and RNA-seq. In every lab, these were the common techniques to understand the expression of particular gene at RNA level. Okay. And proteome, there are several other techniques. 2D gel electrophoresis is one of the famous technique. Every lab still they are using. There are again some immunodetection assays. Then there is some chromatography assay, mass spectrometric assay to study the metabolites. And you can use like multi-top. I think many of you heard what is multi-top. Like that multi-top used to study the protein, it used in the proteomics studies. There are different technologies, and today we are going to deal only with RNA-seq, and I will also give some idea about qRT-PCR, I think. What is QRT-PCR?
Quantification RT-PCR.
Quantitative real-time PCR. Okay? In corona, it is very in corona time. It's very famous technology. Quantitative real-time PCR. Then, like how we can use this one to study the expression of a gene, which is the major
Sir,
dyes are used for RT-PCR. Anyone know? Hey, you're asking something?
sir, RT-PCR meaning real-time PCR, sir, or reverse transcriptase PCR.
There are two different RT-PCR means real-time PCR also there, and reverse transcriptase PCR also there. Reverse transcriptase means quantitative real-time PCR is different and RT-PCR is different. Okay, reverse transcriptase PCR is just nothing but we're converting RNA to DNA by using reverse transcriptase enzyme. Like in mRNA, we can't use the RNA for gene expression analysis. You should convert this mRNA into cDNA. That process is called as reverse transcriptase PCR. Okay. First, you have to convert your RNA to DNA by using reverse transcriptase enzyme. That is called as reverse transcriptase PCR. But quantitative real-time PCR is nothing but at a real time. For example, uh, while you are doing PCR, PCR process is going on, but in screen only the expression is keep on raising. Means on real time basis we can analyze the expression of the particular gene. That's why it is called and also we can quantify the expression. How much, like log 2 fold chain, log 3 fold chain, log 4 fold chain. Like this, we can quantify the expression of a gene, and that too in a real time basis. Means on time only, and time to time we are going to quantify the gene expression while PCR is still running. That's why it is called as quantitative real-time PCR. Okay. Normally, I earlier used to discuss in detail about QRT PCR, but it is not there in the syllabus. That's why I'm not going in detail. But you should study this. Okay. Every lab is using QRT PCR. These geneticists, molecular. Actually, geneticists are more using. Even I also used a QRT PCR for 18 genes. I have done QRT PCR. Okay. Then,
Sir,
yeah.
sir, how can it give the real-time gene expression, sir? Because it's in vivo, no, sir. In vivo also genes work like that.
No. It is in vitro.
In vitro. I think. I'm sorry. In vitro,
See,
I mean.
See that real time that I'm not telling in plant expression. I'm not telling real time means while doing PCR only we are getting uh, like normal PCR. What it will be? You have to wait for the PCR completed. Then you take the PCR solution. Then you have to load into gels. Then you have to see the bands. It is after band. Okay, we are saying okay, the band is there. Okay, gene is there, that time you're telling. But you are not going to quantify here and also not real time basis. After PCR only, in gel we can analyze, okay, after end of the process we are getting. But when the process is still there, we can able to, in screen we can able to see. Okay? That's why it is called as real time. That is implant expression, that is different. That can be done through your GFP protein, implant expression. Green fluorescent protein or luciferase protein. There are several proteins are used. These are actually when the gene is expressing in the cell, it will the fluorescence will going to increase. We can by using confocal microscopy we can study that expression. That is another thing. Implant expression. The plant itself we are getting expression. This is while doing only we are getting we are able to quantify the expression. That's why it is called as real-time PCR. Okay, you got the point?
Yeah.
Then we will jump into the RNA-seq. What is RNA-seq? It's a genome wide. We can study like in QRT-PCR. What will happen? Max to max, how many genes you will get? 50 genes, 60 genes, 100 genes at a time. That too, it's very difficult. at one single PCR plate, max to max, you can go with 18 genes or 10 genes or 15 genes, that much. But you want to seek, analyze how gene expressed throughout the genome at particular point of time. That is called as studying the expression at the genome-wide scale. That is possible by RNA-seq technology. Not only RNA-seq, it is mostly used. There are other technologies also like serial analysis of gene expression and chippers like microarray assay also we can use it for genome-wide expression analysis. But nowadays majorly used is one is chip assay like microarray assay, or we can use RNA-seq. But nowadays that microarray analysis also nobody is doing. The transcriptomics nowadays it become RNA-seq. RNA-seq is nothing but the process of analyzing RNA content and composition of particular sample using high throughput sequencing technology. We already discussed what is the different technologies are available. Generally used are Illumina based platforms to analyze the RNA content and composition of the particular sample. The analysis pipeline, when you look at the analysis pipeline, first we need to map. Now, what is the what is mapping? Means you having your reads, you are mapping these reads into reference genome. This is the reference genome. We are mapping reads based on the sequence information. We are mapping the reads. We are aligning the reads to the reference genome. That is called as mapping. After alignment, you have to quantify how many reads for each exon. For example, the expressed part is only exon. For example, your exon size is for 2 kb. Like how many reads are there per exon? We need to quantify first. Then we have to normalize the data that we will see later. Then after that, you have to do differential expression of the gene. Then you have to visualize. We will go one by one. First, you have to set up the experimental design to do RNA-seq. Like it's my experiment only. Like for example, you're having two parents. One is resistant parent and another is susceptible parent. Your objective is to do after infection of some pathogen, how your gene is differentially expressing. In the resistant parent also, after infection of some pathogen, how your genes are differentially expressed. Then how we can do? First you isolate RNA from each sample, infected, uninfected, infected, uninfected. Then you go for sequencing. Then there is a one particular pipeline. There are several pipelines. Generally use tuxedo pipeline or any other pipeline you can use to identify the differentially expressed genes. Normally you have to go for 6 biological replicates. Without biological replicates, we can't do RNA-seq. At least 2 you can have to do. Then what are the tools we have used for analyzing RNA-seq here? First, you have to millions of reads were there. You have to demultiplex. Then you can go quality control by fast QC. Yesterday, practically in practical, I have shown how to use fast QC to check the quality of the bases. Then after that, you have to go for alignment. For alignment, there are different tools are used. Either you can use top hat, either you can use BW aligner or bow tie aligner, or nowadays we are using high sat. Normally I used high sat. Okay. Then this is for alignment. This is for quality check. And after alignment, you have to quantify the gene expression. For quantification, which softwares generally we use? Cufflink, Dexec, Miso, this are now I used actually another different software called as feature counts. I used feature counts. This is one very easy software, feature counts, to quantify the gene expression. Then another one is called a string type. You can use any of the software, either string type, feature counts, or bow tie, BW aligner, or top hat to quantify the gene expression. After quantification, then what you have to do? You have to compare the expression of infected sample with expression of uninfected sample. To comparison, there are several softwares. which used R environment to do dissect, or nowadays EDGAR is used, most commonly used this dissect, differentially expressed seq, or nowadays like recent papers they are using EDGAR, like TMM based normalization. And for normalization we can do by FPKM, RPKM, or median count. Normally I use median count. Then TMM also you can use. I know this will be take time to understand, but you just remember what is this, okay? You just remember important points. Uh,
Sir, what is pipeline means, sir?
See, this is pipeline. This is pipeline. See, first first you had to sequence, then you had to do quality check, then you had to do alignment, then you had to do quantification. See, this is the pipeline. First you had to do mapping, quantification, normalization, differential expression, and visualization. This is the pipeline actually. For mapping, which tools are we are using generally? We discussed now. Which tools are generally used? Bowtie, ISAT 2, BW aligner. For DNA sequencing, BW aligner, or just remember, okay, if anyone asks you in the exam, you just write. For mapping, for alignment, you just take alignment. Aligning your reads to reference genome, use bowtie or BWA or top hat or ISAT 2, anything you can use for mapping and alignment. Then you have to quantify. For example, this is an exam. This is an exam. How many reads are mapped for this exam? You have to quantify. Like transcript abundance. These are called as transcript. How much transcript are there for each exam? You have to quantify. For that quantification, we use software called as cufflink, string type, string type, STRH, string type, or you can use feature counts. Don't remember, but just I'm telling. Okay? But one, these are the things you know, or if you don't know, it is not, it's not that much matter. But wait, I had to share one another paper. My slides are visible.
Yes, sir.
PPT slides are results. It means it's in not in presentation mode.
Wait. Okay, now this slide is visible.
Yes, sir.
Hello. Yeah. See, first we have to generate FASTQ files from raw reads. Then we have to align this to reference gene. For alignment, we having bowtie, BWA, top hat, shell map, star, or ISAT. There are n number of softwares from different companies are there. Then you have to remember either if you remember these softwares are it is not important. First, you have to general pipeline. You have to remember. First, there will be FASTQ file. FASTQ file for raw reads. Then after alignment, after aligning your reads to reference genome, for see, for example, this is For example, these are the reads. After this is reference genome, this is reference genome. This is each exon. See, this is exon, this is exon, this is exon, this is exon, this is exon. For each exon, you have to align the reads. For example, this read, how many reads are there for that exon? For
Three,
this exon, how many reads are there?
three,
First
sir.
exon, 3.
Three.
Then for this one is 2. Then this one is 3.
Three.
For this exon is 2. This one is 3. This one is 3. Like that, we had to count how many number of reads for each exon in the reference gene. There again, there was just simplify in a simple manner. I'm telling it is not simple as like I'm telling. It is again there is a splice variants are there. We had to identify the exon and intron junctions. Okay, like 5 prime splice site, 3 prime splice site. We have to identify that. Is there any alternative variants? Like after alternative splicing, a single gene give two mRNAs. Try to identify any splice variants are there. That is a very difficult task. But in a simple way, you just understand what is quantification means. We're just quantifying how many number of reads per each exam. That is called as quantification. After alignment, for example, after machine will run raw reads, after generating raw reads, the files which are we store the raw reads in FastQ files. Yesterday I has shown what is FastQ file. After alignment, there are one file will generate. That is called as SAM file, sequence alignment file. SAM file. Mean this SAM file is readable form. Means we can understand, we can read that SAM files and we can understand that what is there in that file. But they are very, very large in nature. That is the way we need to convert into. We will see how SAM files look.
Some people are asking link class,
I shared the link.
not in the group, sir.
Where I shared then. Oh God. Some other group I shared. No, nobody is asking. In group, nobody texted, I think.
Not in group, sir. Some friends.
Okay, okay. They're interested to join course.
No, sir. They already took, but he
Then
messaged me personally, not in the group.
we'll share. First, I will open this. Okay, let it for today's class. Again, I have to share. I'm opening some files. Um, yeah, I'll share my screen. See, actually, this is SAM file. Actually, look like this. It's very big file. So this is SAM files. Here we having after alignment, we having a chromosome number, then the information. See here, like for aligner I used VWA. Then there is a this is the chromosome number NC0 tiny one. Then this is actually then it is sequence. See for each that this one how much length. This is how sample. It's a readable form. You can understand this form, but it is too large. See, this is nearly the SAM file. You can see what I opened. It's more than 1.5 GB. Means there are several samples we're going to generate for each sample. But that's why this is, even though it's readable, it is not that much compatible to use in the further downstream analysis. That's why we need to convert this into BAM file. See, BAM files are very, very short. See here the BAM files, see the same file, there is a BAM, just 666 KB. If you look the BAM files, they are not readable. They are machine readable form, not human readable. What is binary? BAM files means binary alignment files. Binary means just having two numbers. 0101 means it converts the information into binary form, 0101 form. Then it is used for further analysis, BAM file. You just remember, it is asked many times in the exam. What is SAM? What is BAM? You're seeing my screen.
Yes, sir. Yes, sir.
Yes, sir.
They're very large files. That's why. Yeah. This is a binary. See, the all information converted into binary format. 0100. So these are all binary file. We can't understand this. While other file I showed, we can see what is the nucleotides, which chromosome it's aligning. That is SAM file. Means sequence alignment file. But it is a binary alignment mapping. Sequence alignment mapping, binary alignment mapping. BAM files are very shorter. It is only machine readable. It is not human readable, but it is very short. The size of these files are very short, only like some KBs, but it is in GBs. SAM files are it is GBs based on the sequence you are provided for alignment. That's why normally we use BAM files for the downstream analysis because of their smaller sizes. You should remember, raw reads will be stored in faster QCs. After alignment, BAM files will be BAM files will SAM or BAM files will be generated. Sequence alignment map. SAM means BAM means binary alignment map. Then after then you can use these files for further analysis. Either you can use any of the softwares, cufflink, cuffdiff, array string tie, any of the software to quantify the gene expression. Okay. Then
Sir.
how we can do? How we can proceed?
Sir.
Yeah.
I saw some other file BAI file, not BAM file in that list.
Where?
In that the files, sir. You showed files, na? In
There are many other files. Okay.
BA, you
For
showed BAI file, sir, not BAM file. That's why I'm asking.
no,
That is
actually, na
BA.
that bam.ba I store. Actually, why I store? Maybe there is different extension I given. This is bam only. Okay.
Oh, okay, sir.
Actually, bam without sorting. After that, we have to sort. Then it will give an extension as bam. Okay. See the full form
Okay, sir.
bam.bai. There is in Linux command while using. There are different file formats will come. We have to convert it again. We have to sort it. We have to give different output name. There are several things will be there. Then you're seeing my screen like a PPT.
Yes, sir.
There are several softwares used, but normally use Tuxedo. Or they can use any of the software, Top Hat, Cufflink, Cuffmerge, Cuffdiff. Don't remember all those things. Nobody is going to ask, but you should remember raw reads, FASTQ files, alignment files stored in SAM or BAM files. Then for transcript abundance. This much if you remember, it is more than enough. Understood what is the pipeline of RNA-seq? First, you have to map, align, quantify, gene expression analysis. then how data will look like? Then before going to that, well, mapping there, yesterday you were asking like what is met per distance and what is the problem of this one. For example, you're having insert size. What is insert means? Your fragment is there. After aligning, after adding adapter, total it is called as insert. Your this is the fragment. You're adding an adapter for this. Then it will become total insert. Then we are starting while sequencing. Either you can go with single end or paired end. Well, single end reading is just reading in a one direction. But generally, we nowadays we are going for paired end. For example, I told you what is read length. If read length, for example, you take read length is 80. Here opposite read is 80. But your insert size you take 200. I'm just taking an arbitrary example for your understanding. Total insert size is 200 and your read 1 is 80 and read 2 is 80. Then you have to minus total total fragment size minus total read length 80 plus 80. Or you can take anything. Either your fragment size is 400, your read length is 150, 150 plus 150. Means you are getting any value in positive means there is a gap. Means this portion of the gene, like this portion of the fragment, will not going to sequenced because the only read is going to sequenced. Okay, this is during amplification. Up to the here it will going to sequence, up to here it will going to sequence. But who will sequence this middle portion? That is why whenever doing, for example, you take this example, when the read length is 120 and this opposite also 120, means 200 minus 120 plus 120, we are getting minus 40. Means you are getting total, this one is the this is called as met per distance. The inner met per distance, you are getting in minus means you are having overlapping reads. These two reads, blue color, they are overlapping. But here in above side, this is called as gap end reads. They are not overlapping. They are gap end reads. That's why normally for better sequencing, like overlapping reads are better or gaps are better.
Overlapping better, sir, I
Overlapping
think.
is better. Okay, you understood this concept?
Yes, sir. Sir, we should remove that overlapping, na, sir. How can we remove that?
See, why are you going to remove? While aligning, it will be adjusted. While
Uh,
alignment, after
hmm.
alignment, it will get adjusted. Okay? Then there are a lot of things are there. Actually, this is my credit seminar. Actually. That's why I'm very in detail about this. Like it's a Udel second BSRC. I was done. Okay. Then what is functional? Like after DE analysis, you have to go for functional interpretation. Means you can go DG. While this is called as volcano plot. Like this is just look like a volcano. Means how much DG expression. For example, you can see here DG is expressed at -7 also, -8 also. It is upregulated, downregulated, which are in baseline. This is a MACBAN analysis. We can analyze. See, this is biotic stress pathway, hormonal signal. We can visualize the data in many pathways. This is called as enrichment pathway, geoanalysis. It is not needed for you guys. That's why I'm not going in detail. But you just remember what is the general protocol of sequence. PATQ files, SAM files, BAM files, and the general pipeline of RNA-seq. Mapping, quantification, normalization, differential expression, visualization. Normalization, it takes a lot of time to understand. I'm not going in detail. In PhD, we're going to learn. But you just remember the pipeline. Just you remember the this concept it asked in the exam many times. Okay? You take what are the applications? You have to use this one for differential experiment analysis. You can go for transcription profiling. Either you can identify splice variants. Either it can identify the novel genes, either it can like examine intron boundaries we can specify for a particular gene. These are different application. It's unlimited. Okay, and SNP finding. This is the one advantage of RNS. Okay. Then we'll take some questions so that you'll get what are the questions you're going to get in the exam. I have some questions. Yeah. For example, in RNA-seq library, we have 400 base fragments and we did pair-end sequencing of 100 base pair run. Then what will be the mate inner distance? Can you guess? What will be the answer? Now only we discussed.
200, sir. Plus
200
200.
plus or minus?
Plus 200.
Yeah. Means 400 minus 100 plus 100. We are going paired end and each read having 100. It's 200. It's plus 200. Then means they are gap or overlap.
It's gap, sir. Gap.
Yeah. See, this was asked in the SRF 2023. Then, in RNA-seq analysis, the raw sequence data is stored in SAM or BAM files. In
False
RNA-seq
experiment.
analysis, mapped reads stored in FASTQ files. So this should be vice versa. Raw reads are stored in FASTQ files and mapped reads stored in SAM or BAM files. Okay? Then, Oxford Nanopore sequencing platform not recommended for gene expression analysis. And short read sequencing is considered good for RNA-seq analysis. And this also explained yesterday.
First one is true, sir, I think.
Yeah, both are true. Because when you have a long read, I told like we are counting number of reads per exam. If there is a long read, it's very difficult to count. Okay. That's why normally we can use nowadays you can also use, but generally this short read sequencing are more preferred for RNA sequence. That you should remember. Then we'll come to reference genome. Rice reference genome is
Nippon
conver. This is Japonica or Javanica or Indica.
Japonica, sir.
Japonica. Then wheat,
Chinese Springs.
Chinese spring. Maize, B73.
B73. Pigeon P. Asha.
TDS frontier. Pigeon P. Asha. Okay. Then just answer this question discussed in detail yesterday. The example I told.
R is false. Reason is not correct, sir.
During DNA sequencing, multiplexing of samples can be done in a one lane or a flow cell using barcodes. That can be done. Then, okay, I didn't told barcoding. Sorry. While adapter sequence, while adding adapter, see where one flow cell, where can reading millions of fragments, but it will going to mix. But how do you separate? That's why we need to add barcodes. Means it is a short 8 to 10 nucleotide sequence. It's like a signature sequence. Can you heard about signature sequence? Means having a unique signature. Means for example, this signature is added to the adapter. Even though you mix in a millions of fragments, you can able to isolate this fragment by using this signature. That is called as barcoding. Barcoding used for if you are barcoding this, you can mix with anyone because you are able to separate after mixing. That's why the barcoding is done. It is called as multiplexing. In a single go, we can run many samples. We can mix many samples, and in a single run, we can go for sequencing. That is called as multiplexing. Multiplexing done through your barcoding. This is correct. DNA sequencing, where multiplexing of samples can be done in one lane or flow cell using barcodes. It's exactly right. Then Fred score quality 20 means the probability of incorrect base is 1 in 1000. It is wrong. It should be 1 in
Hundreds.
100. Okay. Then structural genomics include genome sequencing and mapping, DNA array and chips, chromatography and mass spectrometry, and qRT-PCR and RNA.
One genome sequencing and map.
The star are functional genome. This is what I want to discuss today.
Sir, once
Hope
go to the second objective you showed.
what?
Second objective you showed, na sir?
Where? Which
Second
side?
one, that BAM files is BAM files and SAM files. That question.
Yeah, this one. RNA cross sequence stored in this one. No?
Yes, sir. Yes, sir. Once summarize that pipeline and the files we get in the after completing of that pipeline procedures.
Yeah, this one only. First, we have a raw sequence stored in FastQ. Then, after reads are aligned to reference genome, we are going to get. Wait. First, reads were there. If it is stored in a file, it is called as FastQ file. Then use this reads and align to reference genome. Then after alignment, you will get one file that is called as SAM or BAM file. First, you will get SAM file that is readable, but it's very large. We had to convert into that BAM file. It's very small, but it is only machine readable, not human readable. Then you also get GTF file. It is in GTF file. GTF means gene transfer format. Means you're having a gene column, chromosome column, this column. That can be used for your analysis, further analysis, counting. It's having exon information. That's why we are able to count. Then after assembly, then we having count rates. Means transcript abundance. Either cuffdiff or cufflink, you can use anything. Then after that, we are going to get a final DEG file in Excel sheet. Okay. Now you got.
Okay, sir. Final DG
Earlier,
file
I
is
used to tell everything about RNA-seq, like how I did analysis, like how Excel sheet look like, how I have to do enrichment analysis. But now, like it is too difficult to understand. That's why I'm not going to confuse you. Now, up to this, understood?
yes, sir.
Any
I
doubts?
understood. The DG files is final file, na sir. After
Yeah, DEG is
uh,
final files.
sir, up
DEG
to
is final
align,
files.
sir. In mapping, after mapping, what file we will get, sir? That FastQ file or
SAM
SAM
and
file
first you will get SAM file.
after mapping or
Yeah.
quantification.
Then, after SAM we will get. Then you have to convert SAM file into BAM files. Okay. After BAM files, you will get based on after BAM files, you will get count matrix file, read counts file. I told now how many number of see after alignment you are going to get how many number of reads per exon you are going to calculate. This is called as count file. It's a simple text file you are going to get count matrix file. This count matrix file for each sample used for DEG analysis. See, okay, understood. You will get after BAM, after quantification, you will get count matrix file. Then that count matrix file is used for differential expression analysis that you will going to get in Excel sheets. Okay.
Okay.
Sir,
One day we will take some complete practical analysis, but I don't have
sir,
yeah,
sir,
yeah.
sir, can you kindly suggest any reference source for sequencing experts?
See, there is nothing like that. I didn't read anything. Okay, just I followed YouTube because I done now. That's why these things I never referred any books or these things. Only research papers. You can read any review paper regarding NGS, There are so many review papers are available. Okay? Where you still rely on books, you just rely on papers. They're all scholars. Okay? I
Okay,
read
sir.
from papers actually. There are so many review papers are there for each technology. For RNA-seq, so many review papers are there. These are all from an excellent review paper is there. I will share. These are all photos I have taken from there only.
Sir,
It's
AKCing
like
and BDCing
a
is enough
toolkit.
for SRF, na, sir?
Yeah, I can't say, but I have to be updated with the recent things. Okay?
Sir, because these questions, what they asked in SRF, they are not
This is
in
asked,
BDC
na?
and AKC, so that's why.
RNA. This is not covered in BDC. It is like nowadays, currently, you should observe the things. Okay, nowadays, what's going in your field? For example, now see in MSc, I did transcriptomics, QTL mapping. Like that, normally, just generally, except most of the MSc students, they are doing this. Has become a simple technique nowadays. Everyone is doing transcriptomics in MSc. My MSc, all two guidemates done transcriptomics only. One did Jivas. Like that, these mapping things are become in part and parcel of MSc research of genetics and plant breeding. That's why you should know about these things. Okay? And reading these things, any like you have to go for syllabus. See, this RNA is specially mentioned in the syllabus. That's why I'm covering. See that you have to just go syllabus. What there in the syllabus, each word you type in the Google. Okay, you will get some information. Now ChatGPT is there. If you put that word in the ChatGPT, you will get at least some information. That is more than enough. For each except standard books, which are the techniques or terminologies available in the syllabus, you just Google it or put in ChatGPT, whatever the information you're going to get, that is more than enough. Apart from standard books. That's why each and every word of syllabus is very, very important. But you can read any papers. There is no special books for these things. Any doubts apart from this? Yeah, I know lots of doubts will be there. When you do not, then you will go into it. Just to
Okay, sir.
remember what is the general pipeline, the previous year questions, observe. And nowadays they left asking these things also. They're more focusing on plan bidding. That's why I'm more focused on plan bidding, basic plan bidding. Any doubts apart from this?
No, sir.
No, sir.
Bye bye. We'll see tomorrow reverse genetics and finish up the unit 5.
Yes, sir.
What to discuss today? Okay, bye bye. Good night.
Good night, sir.